Alrighty, so this is the lecture for Chapter 3 over biological psychology, or as you'll see it commonly abbreviated, Bio Psych or biopsychology. So I just want to preface this lecture by saying this chapter is outside of my wheelhouse, but I'm going to do my best to do it justice. And so in biopsych, we recognize that our brain is the most complicated structure in the known universe. We still don't fully understand how everything in the brain works. In recent decades, biological psychologists have made leaps and bounds in teaching us what our brains are capable of. And then, so basically the brain controls everything you do. So your brain is basically piloting a meat mech suit, as per the meme right here. So nerve cells, these comprise the entire nervous system, both central and peripheral. So a neuron is a nerve cell specialized for communication. This picture shows two nerve cells. So this yellow part and this white part is one, and then this yellow part and this white part is the second. There are 10 times as many neurons in our brain as there are people on Earth, if that gives you any idea of how many there are. It's got a couple different little sections here. So this is the cell body, also called the soma. It controls pretty much what the entire neuron does. It's got a similar composition as cells that you learn about in biology in high school or in college. It's got the nucleus, it's got the mitochondria, the whole shebang. And the cell body is where proteins are manufactured, and it also renews cell components. So basically, it helps keep everything in stock and keep it supplied. So the dendrites, which are these little spindly dealies, receive signals from other neurons. So this that's shown right here, where this neuron's dendrites are receiving a signal from this neuron. This is the axon. This whole white part is the axon. That's the portion of the cell that sends signals to other neurons. As you can see here, the first neuron is sending a signal to the second. And then there's what's called the synaptic vesicle, which is basically a little bubble carrying neurotransmitters that gets sent from the cell body down the axon to the synapse and the dendrites of the next cell. And then when it reaches the dendrites and the synapse, it bursts, releasing neurotransmitters. So what are neurotransmitters, you may ask? They're the chemical messengers used by our body and our brain to send signals between neurons. So synaptic vesicles can be thought of as like liquid gel capsules, like for medicine. So when you take the pill, it's all just in one little sealed thing. And then when it reaches your digestive system, it bursts, releasing what's inside, and that's essentially how the synaptic vesicle works with neurotransmitters. Excuse me. So the synapse, which is this little part, is the space between two connecting neurons. So it's the space between the axon of the first neuron and the dendrite of the second. And the synaptic cleft is a little space in which neurotransmitters are released from one cell to the next. So glial cells are another type of neuron. Basically, they're a cell in the nervous system that plays a role in the formation of myelin and the blood-brain barrier. They respond to injury, remove debris, and enhance learning and memory. So these little kind of orangish red areas on the axon, that's called the myelin sheath. Every neuron has one. Myelin is important for reasons I will get to very shortly, but just kind of keep that in mind. This is the area we're talking about. So The most common forms of glial cells are astrocytes. A single astrocyte interacts with as many as 300,000 to 1 million neurons. You know, that sounds like a big number, but when you consider how many neurons we have in our entire body, that's really not too wild. They communicate closely with neurons, increasing the accuracy of their transmission, help to control blood flow in the brain, and play a vital role in the development of the embryo. So, in pregnant women, these help develop the embryo further along into later stages. Another type is this word that I don't even know how to pronounce it. I'm not going to try. Y'all give it your best shot. But that's another type of glial cell. And this one promotes new connections among nerve cells and releases chemicals to aid in healing. These cells can also produce myelin. So again, the myelin sheath right here, which is a protective coating that insulates the axon. So like many insulators in electricity or electrical systems, the insulation helps the electric message travel down the axon quicker and more efficiently. So as the myelin degrades, those signals kind of get lost in translation between neurons. And myelin is really important because without it, cells just won't work properly. Demyelination, it has been tied to multiple different illnesses, the most predominant one being multiple sclerosis. This is where the immune system attacks the myelin on neurons, causing communication problems between the brain and the rest of your body. So again, demyelination, not good. That's why these cells are very important. And then, a little side note, little gaps right here in the myelin sheath. Oh, that was a terrible arrow. They're called nodes, which help conduct electricity a little bit better. So basically, electricity kind of leapfrogs. So electrifying thought. So again, our brain is basically a giant computer. That's the best way to think about it because of how electrically powered it is. So the neurons respond to neurotransmitters by generating electricity. So that would happen about right here. The brain receives, or the neuron receives the stimulus, which would be the neurotransmitter. It generates the electricity, which is step 2. And this electricity has been recorded in research using electrodes, which allow researchers to measure potential difference in electrical charges inside versus outside of the neuron. When no neurotransmitters are present, the membrane of the neuron is at its resting potential, which is both the before the stimulus and after the stimulus is faded. And this is the resting potential is the electrical difference across the neural membrane when the neuron is not being stimulated or inhibited. This is essentially the baseline state, and the negative charges inside the neuron. Oh, there's more negative charges inside the neuron than outside the neuron. And while at rest, both positively and negatively charged particles flow in and out of the neuron. When the charge inside the neuron reaches a high enough level relative to the outside, so when it reaches this threshold, an electrical impulse called an action potential is triggered. So that would be this part, and then you reach your action potential. Oh God, my drawing. Ooh. And an action potential is electrical impulses that travel down the axon, triggering the release of neurotransmitters. This is the universal language of how neurons communicate. Action potentials are abrupt waves of electrical discharge triggered by a massive change in charge inside the axon, and this is called firing. So you'll hear the term neurons fired or neurons firing quite a bit, especially in Bio Psych and neuropsych. So this is what they're referring to. And neurons either fire or they don't. There's no in between. They don't kind of fire, they don't halfway fire, either they do or they don't. And during an action potential, positively charged particles flow rapidly into the axon, and they flow out just as rapidly. So this spike and decline happens very, very quickly. And these shifts produce a release of electricity, which releases neurons into the synapse. And then an absolute refractory period, which is this big word. So neurons can fire up to 100 to 1,000 times a second. So like I said, this spike and decline happens very, very rapidly. And unlike most extraordinary claims, this one's backed up with peer-reviewed research. Like, they have measured this. They've actually gotten really good at capturing this rapid spike and decline. And each action potential is followed by an absolute refractory period, which you can't totally see it, but right here behind this corner, this is your absolute refractory period. This is the time in which the neuron cannot fire again. So it dips, hits this baseline, stays down, and then when it comes back up right here at 5, that is when the absolute refractory period is over and the neuron is able to fire again. And this becomes a limiting factor in longer axons, but not quite as much so in shorter axons. So like if neurons have a longer axon, they have a longer absolute refractory period than those with shorter axons. So chemical communication. This really breaks down the process of neurotransmitters and how they work exactly. So again, we've talked about what a neurotransmitter is. It's a chemical used by neurons to communicate with other neurons. So electrical events transfer information within a neuron. Chemical events, such as neurotransmitters, communicate across neurons. So between neuron 1 and neuron 2. So after being released into the synapse, neurotransmitters bind with receptor sites along the dendrites of neighboring neurons. So as I mentioned earlier, this is synaptic vesicle. The little dots in here are neurotransmitters. So when this reaches down here, the neurotransmitters are released. These are the receptor sites on the neighboring neurons, and this is the synapse, or this whole thing is the synapse, and then this little part right here is the synaptic gap. So there are multiple different kinds of neurotransmitters. Each one kind of serves their own specific purpose. Think of it like a key unlocking a lock. Like, you can't use your car key to unlock your house, just like you can't use your house key to unlock your car. Neurotransmitters, while their intention is to cross the synaptic gap, sometimes they can be stopped by reuptake, which is where the site where they were released kind of reabsorbs them, so they don't quite make it across. This can prevent too much of a neurotransmitter from making it across the synapse, but it can also lead to mental illness. So while, yes, you don't want too much dopamine, too much serotonin, too much acetylcholine, this can have negative effects as well. For example, SSRIs, or selective serotonin reuptake inhibitors, are often prescribed to treat depression and anxiety, because those with depression and anxiety, their brains reuptake too much of the serotonin, and so not enough is making it across, and that's what leads to depression and anxiety. And different neurotransmitters send different signals. because they play different roles. So some in happiness, some in pain perception. So scientists have developed specific medications to target production or inhibition of certain neurotransmitters. And drugs that interact with neurotransmitters are called psychoactive. Knowing how this interaction occurs can help us predict how certain drugs will affect us psychologically. So, opiates, for example, like codeine and morphine, are agonists, meaning they increase receptor site activity. Tranquilizers, by reducing receptor site activity, they reduce our emotional response to pain, which mimics endorphins. Tranquilizers like Xanax diminish anxiety by stimulating GABA receptors, driving down neuronal activity. And I will explain what GABA is here shortly. It's a super long word. It's easier just to call it GABA. And some drugs like SSRIs block the reuptake of neurotransmitters, especially serotonin. This is the most common reuptake inhibitor medication. Some antidepressants like Prozac cause the neurotransmitter to remain in the synapse longer, so it works similar to an SSRI in that it makes sure more of it gets across the synapse, but it doesn't prevent reuptake per se. And this enhances the effect of the neurotransmitter on receptor sites. And some drugs work the opposite way, and these are called antagonists, meaning they decrease receptor site activity. Essentially, they act as fake neurotransmitters and fool the receptors into thinking the medication is dopamine without exerting the effect of dopamine. So again, I have stolen from the book's table. Oh, damn it. Alright, so again, this is a table found in the chapter. It goes over the main classes of neurotransmitters and their major functional roles. So the first one here is glutamate and GABA. And GABA stands for gamma aminobutyric acid. I think I said that right. And both of them are common neurotransmitters in the central nervous system, commonly abbreviated as the CNS, which we will cover in more depth later in this lecture. Neurons in almost every part of the brain area have both of these, both glutamate and GABA. Glutamate rapidly excites neurons, increasing the odds that they will talk with other neurons. The release of glutamate is associated with enhanced learning and memory. However, when glutamate is at an abnormally high level, it can contribute to schizophrenia and other mental disorders because it can be toxic in high doses. As far as GABA, it inhibits neurons, dampening neuronal activity, which is why most anti-anxiety drugs bind to GABA receptors. They tend to suppress overactive brain areas linked to worry. As far as acetylcholine, this plays a role in arousal, selective attention, memory, and sleep. If you've ever taken Benadryl and felt sleepy, that was your acetylcholine. However, in Alzheimer's disease, neurons containing acetylcholine and other neurotransmitters are gradually destroyed. So too low levels of acetylcholine can lead to memory issues, worse attention, and such. As far as the monoamines, these are like norepinephrine, dopamine, serotonin. So dopamine plays a critical role in rewarding experiences that occur when we seek out or anticipate goals. Areas of the brain rich in dopamine can even be activated by a funny joke. And then as far as norepinephrine and serotonin, those activate or deactivate various parts of the brain, influencing arousal and our readiness to respond to stimuli. I don't even know how to say this, but cells containing it bind to the same receptors as THC. So this plays a role in eating, motivation, memory, and sleep, which is why sometimes when you smoke marijuana, you get the munchies, or you get sleepy. And Another thing is what's called state-dependent memory. So for example, if you do an activity or you learn something while under the influence of something, you will remember how to do that activity better when you are under the same influence again. And that's partially contributed to this. And then neuropeptides are short strings of amino acids in the nervous system that act somewhat like neurotransmitters, but tend to be more narrowly specialized in their jobs. So, for example, endorphins are one type, and these help with pain reduction. Human-made opioids, like morphine, exert their effects by hijacking the endorphin system, binding to endorphin receptors. So, neuroplasticity. Plasticity is simply the ability of the nervous system to change. So basically it's flexible. And of course you guys know I'm a cat lady, so here's a little cute kitty picture of a cat stretching. And neuroplasticity varies as we develop. So our brain is the most flexible when we're young, which is why young children can learn multiple languages quicker than adults, and why there's the phrase, you can't teach an old dog new tricks. As we age, our brain becomes less flexible, which makes it difficult to learn new tasks or acquire new skills. And then our network of neurons can change in 4 major ways. Growth of dendrites and neurons, synaptogenesis, which is the formation of new synapses, pruning, which consists of the death of certain neurons and retraction of axons to remove connections that aren't useful. So this is actually our brain making itself more efficient. And as many as 70% of all neurons can die off. I know that sounds scary, but pruning is actually pretty important to our brain. This isn't a bad thing. Our brains can actually process information a lot more efficiently with fewer neurons. Think of it as cars on a highway. You can drive a lot more efficiently when there's fewer cars on the highway than if it's, let's say, rush hour traffic pre-COVID-19. There's actually a theory that autism may be due to inadequate pruning. Again, it's currently still a theory, it's still undergoing testing. And then, last but not least, myelination. So the insulation of axons with a myelin sheath. And we've already covered why that's important. So, our brains change as we learn. This can result in new synapses and potentation. So potentation is the strengthening of existing synaptic connections so that the neurotransmitters released into synapses produce a stronger and more prolonged response from neighboring neurons. And then neuroplasticity can change following injury or degeneration. So the brain and spinal cord can really only regenerate so much following an injury, which is why, let's say, traumatic brain injuries can cause lifelong difficulties. But sometimes other brain regions can take over the former function of the damaged section. So think of it as like if you work in an office and one of your coworkers gets fired, then you and your coworkers that are still there are left picking up the responsibilities that that coworker had. You know, of course, that's not always ideal because sometimes, let's say, it's a branch manager that gets let go. The sales associates won't know how to run the branch, but if it's another sales associate that gets fired, other sales associates can help pick up the slack. And since many neurodegenerative diseases pose big challenges, scientists are trying to find ways of preventing damage and enabling the brain to heal itself. So a few of these, one of them is adult neurogenesis, which is the creation of new neurons in the adult brain. This is still somewhat controversial, but some believe that by triggering neurogenesis, science may be able to someday induce the adult nervous system to heal itself. And again, this is still in the testing phase, so it's not like 100% yet. And regarding the next point on this slide, stem cells, so some of you are probably wondering, Why does she have a picture of Superman on this slide? Well, this is not only Superman, this is the first filmed Superman, played by actor Christopher Reeve, who is a lifelong stem cell advocate. So stem cells, of course, as we all know, are still extremely controversial, and stem cell research is even more controversial. So stem cells are cells often originating in embryos, having the capacity to differentiate into a more specialized cell. But once the cell begins to specialize, the cell type becomes permanent. One example is researchers can implant stem cells directly into the host nervous system, and they will specialize into neurons. And this will help grow and replace damaged cells. They can also be used in gene therapy by providing replacement genes. So why is stem cell research controversial? So advocates champion its potential for treating serious diseases like diabetes, Alzheimer's, and some cancers. It's even been used in treatment of arthritis to help prevent further degradation of the damaged tissue. Whereas opponents believe that the cells used are living beings, even though the cluster of cells created in labs are smaller than a 12-point font period. And if these are the first stage of life or not is outside the realm of science. It is a metaphysical question. We don't truly know when life begins, and there's always going to be different opinions on when it does or when it doesn't. And because of this, people will continue to disagree on if we should do stem cell research or use stem cells. So the brain behavior network. So brain behavior moves through a few steps. Our brain decides to do something. Our nervous system propels the body into action. And then we need to locate and operate our bodies. So our nervous system is a superhighway with two-way traffic, input and output. So it receives input and it also gives output. And sensory input comes into and decisions come out of the central nervous system, which is this diagram, which is part of our nervous system containing the brain and spinal cord that controls mind and behavior. The other part is the peripheral nervous system, which is the nerves in the body that extend outside the central nervous system. So the spinal cord, as we can see in this one, has a bunch of different neurons leading off of it. All the neurons that come off the spinal cord and the brain and connect to the other parts of the body, that's the peripheral nervous system. And the peripheral nervous system is further divided into the somatic nervous system and the autonomic nervous system. And the somatic controls voluntary behavior, and the autonomic controls involuntary behavior. So the best way to differentiate is think of autonomic as automatic. You don't think about your heart beating. You don't think about your breathing. That's the autonomic nervous system. So the central nervous system has 6 distinct sections/systems. The first of, so the brain and spinal cord are protected by meninges, which is 3 thin layers of membranes. Additional protection comes from the cerebral ventricles, which are fluid-filled pockets that extend throughout the brain and spinal cord. If you've ever watched a medical show, Scrubs, House, Grey's Anatomy, and you've heard the term CSF, that stands for cerebral spinal fluid, and it runs through those ventricles, and bathes our brain and spinal cord, providing nutrients and cushioning us against injury. So the main part of the central nervous system, the main part of the brain, is the cerebral cortex. It is the outermost part of the forebrain, responsible for analyzing sensory processing and higher brain function like language and reasoning. So if you touch right here on your forehead, that's directly in front of your cerebral cortex. all right here. This is cerebral cortex. It is the largest component of the cerebrum or forebrain, and that's the most highly developed area of the human brain. It has 12 to 20 billion neurons in humans and accounts for about 40% of the brain's volume. This gives us our advanced intelligence abilities. And fun fact, there's a thing called encephalization quotient that's used as a proxy to estimate intelligence by counting the number of neurons in the forebrain. So for example, elephants have the highest amount of neurons in the forebrain of any mammal, including humans. Manta rays have the highest for any fish, and octopi have the highest of any invertebrate. And the cerebrum has two central hemispheres, as you can see right here in this little diagram. There's the left hemisphere and the right hemisphere, each of which serve distinct yet highly integrated functions. So, for example, the left brain is responsible for analytical thought, detail-oriented perception, ordered sequencing, rational thought, verbal communication, cautiousness, planning, math and science ability, logic, right field vision, and right side motor skills. So for example, left brain controls right arm. And the right side is responsible for intuitive thought, holistic perception, random sequencing, emotional thought, nonverbal communication, being adventurous, being impulsive, creative writing / art abilities, imagination, left field vision, and left side motor skills. So right brain controls left arm. And the two hemispheres are connected with what's known as the corpus callosum, which is a large band of fibers. So if you see the little indentation right here in the middle, that's the corpus callosum. And while this is typically an essential piece of anatomy, cutting it has been used to treat extremely, super extreme cases of epilepsy that don't respond to any other treatment. But of course, like any other medical treatment or any treatment at all involving the brain, this can have adverse side effects or adverse consequences. So as far as the lobes, the cerebral cortex is made up of 4 areas called lobes. We have the frontal lobe, which is this blue area. We have the parietal, which is the yellow. We have the temporal, which is green. And we have the occipital, which is red, pink. And of course, it is important to note, I'm sure you guys probably know, the brain is not actually that color. It's like a weird pinkish gray, mushy color. This is just used to diagram out the different lobes. So, the frontal lobes perform executive functions that coordinate other brain areas, so motor planning, language, memory, as well as they oversee and organize most other brain functions, a process called executive functioning. So think of the frontal lobe as the CEO and the rest of your brain as the corporation. And these are located at the front part of the cerebral cortex. So again, right here, right behind your forehead, that's your frontal lobe. In most brain areas, there's a deep groove called the central sulcus that separates the frontal lobe from the rest of the cortex. So in this little diagram, that thick black line, that would be the central sulcus. So the motor cortex is the part of the frontal lobe responsible for body movement. So if any of you are clumsy like me, you can blame your motor cortex. Each part of the motor cortex controls a specific part of the body. with regions that require more precise movements, like fingers, taking up more space. This is often depicted using a homunculus, which is this weird looking diagram I have for you guys. So if you notice, areas that don't require very precise movement, like legs, shoulders, elbows, stuff like that, they don't take up as much space as your hands or your face, which both require very intricate movements. And in front of the motor cortex, so if you see this little dotted line right here on the diagram, right there, that is your motor cortex, and just in front of that is your prefrontal cortex, which is responsible for thinking, planning, and language. So one of the major regions of the prefrontal cortex is called Broca's area, which is responsible for aiding in speech production. There is actually a disorder called Broca's aphasia, in which people can no longer speak or have extreme difficulty speaking due to damage to Broca's area. This is often seen in people that have experienced strokes or have advanced stages of Alzheimer's. The prefrontal cortex also contributes to mood, personality, and self-awareness. Damage to this area can increase impulsive or criminal behaviors. Damage can result from stroke, blunt force trauma, a traumatic brain injury, etc. Now, while not everyone who receives damage to this area will become compulsive or criminal, it is a contributing factor to those who do become compulsive or criminal following damage to this area. So the most famous case of prefrontal damage was Phineas Gage, who took a railroad spike to the face in 1848, piercing his brain and causing permanent brain damage. He lived, but his entire personality changed. And this was due to the damage done to the prefrontal cortex. So basically, he was filling holes with gunpowder to break up really stubborn rock formations. Essentially, gunpowder went boom, railroad spike went yeet, and the rest is one of the most fascinating contributions to biological psychology. And there's growing historical evidence that Gage's personality change may not have been as radical as it was thought to be for a very long time. The brain is still fairly flexible in adulthood, and some of his injuries may have partially healed. This is based on more recent research. And some of the function may have actually returned. Regardless, Phineas Gage had a major contribution to Bio Psych as we know it. Interesting enough, a second case of this emerged in 2012 in an Argentinian construction worker, Eduardo Leite. While he seems to be doing well, it is still too early to determine if his personality changes will be permanent. So, the parietal lobe. This is the upper middle part of the cerebral cortex, lying behind the frontal lobe, and it's specialized in touch and perception. It also helps us track objects' locations and process others' actions, represent numbers, shapes, and orientation. Because of its role in spatial perception, patients with parietal damage have a hard time making sense of their surroundings. So, and interesting enough, the parietal lobe can be a little bit different across cultures. So what I have here for you is what's called the Mueller-Lyer illusion. And the big thing is, which of these lines is longer? For anyone who grew up in an industrial culture where you're used to square rooms and square buildings, you will say the top line is longer. But they're actually the same length. And interesting enough, people who did not grow up around square rooms or square buildings, they don't have this effect. They recognize both lines are the same length. A common consequence of parietal lobe damage is neglect of the opposite side of the body from where the damage occurred. So as I mentioned with left brain and right brain, left brain contributes to the right side, right brain contributes to the left side. And this is common in multiple lobes. It's not just the parietal lobe. Damage on the left side will be seen in diminished ability on the right side, and vice versa. Another section is the temporal lobe, which is the lower part of the cerebral cortex that plays roles in hearing, understanding language, and memory. This contains the auditory cortex, which is the part of the cortex devoted to hearing. The language area in this lobe is called Wernicke's area. Part of the temporal lobe involved in understanding speech. So, like Broca's area, there is another aphasia called Wernicke's aphasia, and that can result in people not being able to understand speech or have a diminished ability to do so. Again, this is common in stroke patients or those with advanced stages of Alzheimer's. And the lower portion of the temporal lobe is critical to storing memories of autobiographical events, which is commonly referred to as episodic memory. The occipital lobe is the back part of the cerebral cortex specialized for vision. This contains the occipital cortex, responsible for seeing. This is why when you get bonked on the back of the head real hard, you see stars. As far as cortical hierarchies, when info on the outside world is transmitted by a specific sense, it reaches the primary sensory cortex, which is regions of the cerebral cortex that initially process information from the senses. So this is like the first stop on the train line. And after the sensory organ receives the information, it passes it to another area for that sense called the association cortex, which is regions of the cerebral cortex that integrate simpler functions to perform more complex functions.